Hello, everyone. Welcome back to my channel. My name is Dr. Anwar Youssef Dunbar, and this is Big Discussions 76, Science and Technology. First of all, please like this stream, please share it, and please subscribe to my channel. Well, today is April 2nd, 2021, and today I want to talk about my graduate thesis. You guys know I am Dr. Dunbar. That's the name I use in the YouTube streets. But I never talked about what I actually worked on to become Dr. Dunbar. And I thought that it would be educational and very insightful to capture this live and also on the playback for those who want to watch it and listen to it. But I also realized that I've done a lot of we hear what's going on, sheet. I've also realized that I've done a lot of storytelling here. I've done a lot of reporting on the pandemic, how the virus works, what we know about the virus, the different medications, how those medications work, and most recently the vaccines. But I haven't presented a lot of data. And if any of you all, well, I know a lot of my followers here follow the scientist supreme, the lioness, Nicole Ali. Shout out to Nicole Ali. She's presented a lot of data on her channel, and I haven't done a lot of that. And so I wanted to try that. I wanted to show you guys, my viewers, some Western blots and some HPLC chromatograms and some in vitro assays and those kinds of things. So in this stream, I'm going to get a little bit into the woods, or down in the weeds a little bit. And the challenge is going to be not to lose people. I'm going to use some esoteric terms here, and I'm going to use, we're going to venture into some areas that we haven't ventured into on this channel before. But I thought that this was a good time to do it, and I think it's necessary to do this. I want to shout out Sheed, who just gave his official what's the word I'm looking for? His calling card. She gave his calling card. We hear in the last 2 weeks, I found out what it was like to have something taken down by YouTube. I basically, it's called waterfalling, where you take a piece of your live stream, cut it out, and make that a featured video. And I uploaded a little piece of me and Nicole talking about the difference between the Johnson & Johnson vaccines and the Pfizer vaccine and the Moderna vaccine. And it was harmless. I thought it was harmless, and YouTube, I put it up. She was one of the first folks to like it and watch it. And then I came back to my channel about an hour later, and the video was down. And I went over to my Gmail account, and they sent me an email saying this violates the community guidelines. And when I logged back onto this page, I saw the picture of the guy with the whistle saying, this is a warning. If this happens too many times, you won't be able to live stream and blah, 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 blah. So I know what that feels like now, to have something taken down and policed by the YouTube, I think they call it the YouTube Politburo. So shout out to the Roger Report. He's had channels, some content creators have had channels taken down. So that was just one video. So I can only imagine what it's like to have a channel taken down. So in any case, this should be a lot more harmless than talking about the vaccine. So I see Tasha's here. Tasha, thank you for stopping through. Tasha, I saw something over on Instagram. I won't talk about it here, but it surprised me. I was going to ask you why I was going to ask you about that picture, but maybe I'll do it after I get done talking here. So this live stream, this one is about It's a discussion of the first part of my graduate thesis. And if you're viewing, if you're new coming in, please click the like. I was thinking about this. This is as you'll see, this is a complex thing, and the challenge again is going to be to keep it simple and digestible for the laypersons. And I was thinking about putting this off, but I caught the tail end of Roger's discussion yesterday over on the Roger Report. I'm over there pretty much every day with guys like Sheed and Rael and Black Uru and Roger and Mr. Z. And one of the guys saw me come up in the comment section. He said, we got to get him up here. Of course, I've been up on Roger's panels numerous times, but I just thought about the unique skill set and the unique perspectives as a scientist that I bring to this space. And I was thinking about putting this stream off until next week, but something said, what, just go today, do it today, because this is important. This is important to get out. And I think that one of the things I lend to this space is acting as a symbol. And I think Nicole feels the same way about what she's doing. Before I jump in, I just want to, I still have this picture here from my discussion with Officer Charles Faulkner over on my main channel. We talked about the Great Faulkner Initiative. And this is a diagram from the Great Faulkner Initiative. And one of the things that Charles talked about in that live stream with me, you guys can go over to my main channel and check that out, was that the importance of getting, in this instance, the boys into STEM careers. And I think that, yes, we need to get the boys, the younger men, somehow over into the STEM careers. And I think that it's one thing to say that, but I think the how is important, the how and the why. So it's one thing to say you should go pursue biology or analytical chemistry or gene editing or cancer biology or neuroscience. You should go pursue those. But there are a lot of challenges coming from the inner city. I'm from the east side of Buffalo. And as I will describe as I talk about my graduate school thesis, there were a number of challenges, not just with my science project itself, but also learning the culture of science and also leaving some parts of the culture that I grew up with behind, and also continuing to navigate the culture that I came from and the neighborhoods that I came from. So this is very much a journey. My science journey was very much a journey through socioeconomics and that whole strata. I'm going to show you guys a couple of blog posts that I wrote before I jump in. And this is relating to what I just said about the challenges in pursuing my doctoral degree and finishing my and pushing my graduate thesis project through to completion. I published this on my blogging platform a couple of years ago, actually, and it turned out to be pretty popular. This one is called The Story of How I Earned My STEM Degree. So I'm going to show you guys some data today, but I can't emphasize enough that this was a journey. It was a tremendous growth period. And so I talked about, in this blog post, I talk about what my major scientific learning points were. But at the end, I also talk about some of the the challenges outside of labs, some of them coming back to Buffalo on holidays. And one major challenge was the person that I dated during graduate school. So there were a lot of challenges in this process, learning how to do science. I'm also, and I'm going to leave all these in the description box below, but I'm also going to leave this one here because this what I'm going to tell you guys about, where we're getting into the quote unquote the basic sciences. Let's see. And I did not cue that one up. Give me a second here. The basic sciences, basic research. Okay, that's loaded up. There it is. What I'm going to be talking to you guys about today is basic science and basic research. And that's what you do when you're pursuing a PhD in one of the sciences. You're at a workbench, you're conducting experiments, and you are pursuing new knowledge. And I had to learn about this all in real time when I was working on my graduate thesis. That's going to be in the description box below. Actually, I'm just going to go ahead and drop these links in as I go along. Let's see. This one is what are the basic sciences and basic research. And again, this is important because, you can tell younger people to get involved in the sciences, but if you don't know what it is, it could be a very, very nebulous thing. And I'm going to drop this one in the chat right now as well, the story of how I earned my STEM degree. Some people grow up with science, as I'll describe, and then other people, you'll learn it. You have to learn it on your own when you're out pursuing your career. Okay, so I'm going to stop this here. So I've told you all that I have a background in pharmacology. And I've talked about the fact that there's a clinical side of pharmacology, but there's also a research side. So everyone pretty much has experienced the clinical side. You've gone to the doctor, you've gotten sick for some reason as a child or as an adult or as a senior. Perhaps you've had to have a surgery done, and you've had to take pain medication in the recovery, but also before your procedure, they had to put you down. Well, that's all pharmacology. But there's also a research arm. And the research arm is where all of our new drugs come from. It's where all of our new medications come from. It's where our new vaccines come from. And it's also how we start to understand disease states. When I was an undergraduate at Johnson C. Smith University, which is an HBCU, one of my professors, I don't think he's still with us, Dr. Larry Thompson, he was a physiologist. We would sit and talk, number one, about his experiences getting his PhD at the University of Cincinnati. He experienced a lot of racism during his graduate thesis. But he would talk and tell me and say, well, medicine, it used to be gross anatomy. It was all gross anatomy. So just understanding how the body was built and understanding what organs were in the body. But he said, Anwar, now everything for you guys is turning into everything for you guys is turning into molecular biology and biochemistry. So there was a shift as science evolves. There was a shift from understanding medicine through more of a anatomical approach or understanding. And then now we know what genes are. We know how genes work. We have so many assays and biomarkers. I'm sure anyone who's subscribed to Nicole's channel, you've heard her talk about biomarkers, and we have assays and so many research tools and so many diagnostic tools and so many medicines that we didn't have back in the day. So there's a clinical arm of pharmacology and a research arm of pharmacology. So let's jump into my graduate thesis. What did I work on to become Dr. Dunbar? So again, the challenge is going to be to get through this without confusing my audience too much. And if you have questions, if you're going to hang on with me for the duration of this, please feel free to ask questions there in the chat. My plan is to only go for about an hour. So pharmacology, the study of how drugs and xenobiotics interact with the human body. And it's also the study of what we can do to those drugs to make them more effective. So how do you figure out what a drug does? How do you figure out how it works down to the protein and the DNA level, down to the molecular level? It is unethical to perform experiments on humans, though we are moving into an era where you can use human cells in some instances. There was one of the controversies with the Bush 43 administration was using the stem cells. But there are approaches now where you can ask questions in human cells, and I think in human tissues. But in large part, it's unethical to perform experiments on humans. So what you then have to do, and what science was doing for a number of years, was you would use lab animals. Those of you who are sub to Nicole, she does a lot of work with mice. She's probably done quite a bit of work with rats at some point. This is a Sprague-Dawley lab rat. Some of my first experiments were with rats. But here's the caveat, though. Here's the thing. You have to think about complexity. Both this rat is a multicellular organism. We as humans are multicellular organisms. So because of the complexity of the organs, the tissues, and the cells, there are certain questions you can and you cannot ask when you're looking at a multicellular organism. So some questions you can only ask. Give me a second here. Some questions you can only ask and answer using experiments with cells, and some questions you can only ask and answer using proteins. Proteins like this one. I had it earlier. Here. Proteins like this one. This is the ribbon diagram of an enzyme called neuronal nitric oxide synthase. This is the protein that I primarily worked on in graduate school. And when you think about proteins, I describe proteins as the workhorses of the cells. So, and I've recorded a couple of videos on transcription and translation. For simplicity, the DNA from DNA gives rise to RNA, and RNA gives rise to the proteins in our cells that do any number of things. So my graduate advisor, before I came into his research lab, he studied enzymes metabolism, enzymes that metabolize drugs called cytochrome P450s. This is a ribbon diagram, a three-dimensional structure. of a cytochrome P450 enzyme. These are highly expressed in our livers, and they are very important for drug metabolism. So my graduate advisor spent his graduate years studying these, and also his postdoctoral years studying these. And then he became interested in this enzyme, nitric oxide synthase. There are 3 isoforms. It means there are 3 forms of it. One form is involved in immune response. That's the inducible form. And then there are 2 forms that are what we call constitutively expressed in the nervous system, in our circulatory system, and also in penile tissue. And so I'm going to show you guys something here. What my graduate advisor found was that in rats, yeah, you should be able to see that now. Yeah, you can see that now. All the papers I'm going to show you guys are available online. So none of this is top secret. So this paper is entitled The Metabolism-Based Inactivation of Penile Nitric Oxide Synthase Activity by Guanabens. So what does that mean? So metabolism basically means that in this context that an enzyme, in this instance, nitric oxide synthase, it can transform a drug. It can metabolize a drug. It can interact with a drug and give rise to another product or transform the drug into something else. So what he found, and I'm going to try to go through this quickly, he found that this drug here in the middle, Guanabens, that's an antihypertensive drug. What it could do to our enzyme in rats was that it could inhibit the activity. I'm not going to get too much into this, the activity portion, but it could cause the levels of the protein to go away. So if you look at this control bar here, that's a control rat. And then the rats were treated with 5 milligrams per kilogram of the drug Guanabens. So you're seeing a loss of the protein. But this drug over here, N-nitroarginine, did not cause the protein to go away. So one drug caused the protein to lose its activity and also to lose the protein itself. The other drug caused the protein to lose its activity, but did not cause the protein to go away. So that's important. So the question is, why is one drug exerting an effect on the protein that another is not? So what's the difference between the interactions of the drugs with the proteins to give the result? What my graduate advisor then did, and his staff of scientists, what they did then was they approached it another way. So we have this data in the animal. So now we need to use a cell model to start to map out and figure out on the cellular level why this particular thing was happening. So they conducted another set of studies here, this time using a cell model. This paper is entitled Guanabens Mediated Inactivation and Enhanced Proteolytic Degradation of Neural Nitric Oxide Synthase. Okay, that's a mouthful. But what they wanted to do again was on the cellular level, was they wanted to see what the mechanism for that protein loss was. Okay, so what you can do, I'm going to stop here and go over a couple of techniques. So again, so you have a protein that you're interested in the human and the rat. Okay. but you want to study that protein by itself, or you want to study it in a less complex way. Well, one of the things that you can do is you can isolate the DNA from the human or the rat, and you can transfect it. You can insert that DNA into a cell line, okay? And you can ask questions about that protein in this cell line that you can't ask in a rat or in a human. Again, that's called a transfection. So what my graduate advisor and the staff at that time did was they transfected the enzyme into human embryonic kidney cells, and they asked a set of questions. Specifically in the rat, why did the one drug kill the activity and make the protein go away, while the other drug stabilized the levels of the protein but killed the activity? So they conducted these studies here. Once again, you see our drug killed the activity. I'm not going to focus on that at this point. But what they were able to do was they were able to measure the protein in those cell lines. So you have your control, you have your drug here, guanabens, cyclohexamide, that's a protein synthesis inhibitor. You use that with cell lines. What they were able to show was that the drug guanabens caused a loss of the protein here in the cells. So similar to the animal, it was making the protein go away. The same was true for this compound N-methylarginine, which is a substrate analog for this neuronal nitric oxide synthase that was also able to make the protein go away. This drug over here, similar to the animal study, it stabilized the protein to a large degree. So there's something about these two drugs here, the guanabens and the N-methylarginine, that made the protein go away that the N-nitroarginine didn't. So there's a selective mechanism somewhere. They showed similar data here. The guanabens drug made the protein go away. These two drugs didn't. The N-nitroarginine here and the 7-nitroindazole stabilized the protein. So these drugs are doing something unique to the protein. And then this is a time course. Here, you have your control and you're treated, your guanabens treated. And what they saw was that the drug guanabens was able to accelerate the loss of the protein. So this guanabens was doing something to the protein that was making it go away much faster than the control. So what is a clue? What are some clues as to why this was taking place? So if you know that a protein is going away, that suggests that there is a protease involved somewhere. Okay, Rico M. Dr. Osawa is still strong as an advisor in pharmacology. Okay, Rico, are you a student or a postdoc? Or did you pass through the Michigan Department of Pharmacology? As you can see on the paper, Dr. Osawa was my graduate advisor. And yes, he's very brilliant, very driven, and he taught me a lot about this. Not just the science, but also about being a professional. And also, Rico M., thank you for passing through and coming to my live stream here. So the question is, why would this protein go away like this? And what could be the cause? So my advisor and his team of scientists, what they did was they used a set of protease inhibitors, okay? And a protease inhibitor is just that. It inhibits a protease. So it clogs up a protease and stops it from chopping up an enzyme or a substrate. So when you have a protease inhibitor, it's like a fingerprint. You know which protease it's going to inhibit and clog up. So this one, the lactocystin, that's what that LC stands for, and this MG132, these all basically pointed to the 26S proteasome system, okay? And that's what this set of experiments in these cell lines basically told them. So they saw it go away. They saw guanabens accelerate a loss of the protein in the rat. And now in the cell line, they saw it accelerate in the cell line. And with these protease inhibitors, this suggested that it was due to a system called the 26S proteasome. Okay, so I'm going to stop there, take a look at the chat, take a sip of my coffee, and then I'm going to tell you about what I worked on. And I want to thank you all and applaud you all for those of you who are staying on and listening to this. So Rico says, my GF, okay, my girlfriend got a master's in pharmacology recently. Okay. 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 I would ask who that is, but this is YouTube. This is public, so don't you have to say that now. If anyone wants to email me or follow me on social media, this is where you can find me. There's a Big Words blog site, blogging platform. This is the email address that I officially use for YouTube. It's long. Big Discuss 76 at Big Words Are Powerful. There's a Big Words blog site Facebook page. There's a Big Words Are Powerful Twitter account, and that's me on Instagram. So if anyone wants to, so Rico, if you want to email me behind the scenes, for whatever reason, or anyone, that's how you can get in contact with me. So I did not explain what a protease is and why this all matters and how this all fits together. So a protease, so an animal cell, our bodies are made up of billions and billions of these. And depending upon the tissue type, the cells have particular functions and particular properties. But the majority of our cells have a nucleus and a cytosol and a cell membrane and ribosomes and a Golgi apparatus and mitochondria. But also in that cytosol, there are peroxisomes and there are proteases. I don't see any proteases in this cartoon, but what a protease does is it basically cleaves a protein. And so our cells have mechanisms for creating proteins, and then there are housekeeping mechanisms for degrading and cleaving those proteins once they've got to the end of their life cycle and once they have outlived their usefulness. So our cells have mechanisms for creating proteins and mechanisms for degrading proteins. Okay. And those two arms work hand in hand. And it's theorized that we know that when any of those mechanisms break down, when any of the natural mechanisms in the cells break down, that's when disease states start. But when it's been postulated that when certain pathways break down, and for example, when proteins become misfolded or unfolded, and when they build up in cells, that can lead to cancer, and that can lead to disease states. In any case, the thumbnail for this live stream and where my the last paper I showed you concluded was this. This is the 26S proteasome system. So this is and this is from about 15 years ago. So science has evolved, and there may be more known about this now. But this is the 26S proteasome right there in the center. That is it's a protease system. Those purple UBs, those are proteins called ubiquitin. And basically what you need to know is that in order for a protein to be targeted for cleavage and digestion by this proteasome here, that protein has to be tagged with these ubiquitin proteins. And those are tagged by ubiquitinating enzymes represented by E1, E2, and E3. So once they're tagged with those ubiquitins, they are recognized by the proteasome, Deubiquitinating enzymes take the ubiquitin off, and the protein is cleaved into little bits, and the amino acids are recycled so the cell can use them. And this is an ATP-dependent process on the front end and on the back end. So that last paper I showed you suggested that our enzyme, nitric oxide synthase, was degraded or is degraded in the cells by this proteasomal system. Okay. So then the question is, how does this happen in terms of signal? How does the cell know to target our NOS protein to this system? And what did those drugs do selectively to our protein to target it more rapidly to this system? So before I jump into my actual work, I'm going to tell you a little bit about the nitric oxide synthase protein, and then I'm going to tell you about some of the experimental tools that I used during this work I'm going to show you. This is a ribbon diagram of nitric oxide synthase. It functions as a homodimer. That's a common term in biochemistry and enzymology and in cell biology. A homodimer. Homo, we all know what homo means. It means the same. And a dimer means two parts. So you have a protein that has two parts that are the same. If you think about hemoglobin, which carries oxygen and carbon dioxide, that is a tetramer. So it's got 4 identical parts, I believe. So nitric oxide synthase is a homodimer. You break that homodimer up into 2 parts and you have 2 monomers. What does that mean? That means that each of these 2 parts, if you lay them down like straight, I thought I had this queued up and ready to go. There it is. If you lay these down and take a look at them, each of those monomers, just focus on the top one. You have an oxidase domain and a reductase domain. So the oxidase domain possesses a heme group. I'll tell you about what that is in a second. It has a zinc finger, I believe. That's what the zinc is for, where we think that the dimer locks. And then you have a reductase domain, which is the bottom, which are the yellow parts, where the enzyme binds reductase. So the enzyme needs reductase for electrons to catalyze its reaction. In its actual reaction, I don't think I have a cartoon handy here. The actual reaction is that it uses, I'll discuss this in part 2 in detail. The actual substrate for the enzyme is L-arginine, which is an amino acid, which there are, I believe, 21. But the nitric oxide synthases use L-arginine as a substrate, and they catalyze the production of nitric oxide, which is a signaling molecule, which in men that leads to penile erection, and it does other things. And also it forms L-citrulline, which is a byproduct. So that's the enzyme. That's the protease. And before I jump into my graduate research, I'm just going to walk through a few tools that I think are germane to understanding this discussion, because understanding someone's scientific research involves understanding the background, it involves understanding the question being asked, but it also requires understanding the techniques and the methods being used. So I am going to start with an SF9 insect cell expression system. So you have a complex organism like a human or a rat, but you just want to study and characterize one enzyme. Well, how do you do that? What I'm about to show you is analogous. When I was doing this, in my mind, it was analogous to farming. Shout out to Irv Gotti. Farming. It means you're growing something and you're harvesting it. So you're interested in the one enzyme, and this rat has its body contains thousands of enzymes and proteins, but you're interested in just one. What you can do scientifically is you can isolate the DNA sequence for that enzyme that you're interested in. You can insert that into an E. coli. You can grow up that plasmid, and then you can create a virus. It's called a baculovirus, a recombinant baculovirus. I have another comment here. NC Worker, good afternoon, everyone. NC Worker, thank you for stopping through. Once you create that baculovirus, you what you do is you grow an insect cell line, SF9 insect cells, and you infect those cells with the baculovirus, right? And then what happens is after about 2 days, depending upon the titer of the virus, titer is just another word for concentration, you can get those insect cells to express your protein in massive quantities. And that was my job in the lab, in addition to my research project. So it's called an SF9 overexpression system. So you create a virus with the protein of interest, you infect your insect cells, and then you harvest the protein from those cells by busting them open and creating a cytosol. For a visual, so you guys can see what it's like to grow the cells, what you do, the way you do that is you purchase an orbital shaker. This is an orbital shaker from Innova. We had a big one like this, and we had a small one. And those flasks on the inside of that orbital shaker contain medium, and they contain the insect cells. So this spins around. Those cells are spinning around continuously, dividing until you infect them with the virus. And then 2 days later, they become nice and plump. And then you bust those cells open. I won't get into how I did that, but you create a cytosol. And then what you do at that point is you pass that cytosol over a column. In our lab, we did a lot of column chromatography. And all that I'm going to say here is that what you're trying to do is you're using a solid, a stationary phase matrix here represented by the blue. You're putting your cytosol with your protein on over the top, and you're using that stationary phase to isolate purified protein. So that's what we're doing. That's what we did. Then this was one of the basic techniques of my graduate advisor's lab, was generating massive amounts of this protein in vitro so we could study its activity, so we could study its biochemistry, and so we could study how it interacted with other proteins. I didn't tell you guys what a heme is. Basically, if you look at this ribbon diagram, you see two gray centers in those proteins, in those protein bundles. Those are heme groups. Our oxygen has heme groups. Our drug metabolizing enzymes in our liver, those have heme groups. That's where the catalysis of the enzyme takes place. I see another comment here. Tasha said, how cool. Oh, the whole crew's here. Tasha says, how cool. Miss Burdine is here. She says, sounds interesting, Dr. Anwar, but it might take me the rest of the year to understand it. Yeah, Miss Burdine, that's the challenge of presenting this, is making it, is understanding it. It's for the layperson to understand it. She says, of course, I'm late to the presentation of the video. Okay. So that was one of our bread and butter techniques, was using insect cell systems to create massive amounts of this protein so we could study it in vitro, its activity, its structure, and how it interacted with other enzymes and other proteins within the cell. So at this point, I'm going to, and again, when you think about this, think about the concept of farming, because we were literally farming this protein. We were making it, growing it, harvesting it, and then using it for a purpose. Okay, so interacting with other proteins. Can anyone, I'm going to tell a joke here. Can anyone in the chat tell me, there was a comedian in the 80s who made a joke about test tube babies? Does anyone know who that is? Look at the chat right now. There was a joke. There was a comedian. He passed away, but he made a joke about test tube babies. I'm going to take a sip of my coffee here and see if anyone gets it. Okay, I'll give you a hint. This comedian became famous off a set of jokes about a woman's kids. No, not Red Fox. That's kind of close. He was a darker skinned guy. Anyone? Very funny, very creative. Arguably died before his time, like Bernie Mac. I'll take a sip of my coffee and I'll wait another minute. Moisten my throat. Miss Burdine and Tasha, I would think you guys would know this. A comedian from the 80s and the 90s. Before he died, he was in movies. And his signature joke was about a woman's kids. And this woman he talked about was the avatar for this type of woman throughout the Black community. All right, everyone. I'll wait 10 more seconds. 1001, 1002, 1003, 1004, 1005, 1006, 1007. Yep, 1008, 1009, 1010. Who was the comedian, though? Anyone? All right, I'll say it. Robin Harris. Yep. Them is Bebe's kids. You know, we don't die, we multiply. But one of Robin Harris's other signature jokes was test tube baby. His comedy was brilliant because he was always ribbing on people. But one of his other signature jokes was about people being test tube babies. Does anyone know what? Well, let's do some Q&A here. What kind of pregnancy is that when someone is fertile? Yeah, what kind of pregnancy is that when someone is a test tube baby? Let me see if you guys know that. It's a specific kind of pregnancy when someone is a test tube baby. It begins with an I. Yep, conscious cop, very good. That's an in vitro fertilization. Surrogacy, no. Well, Tasha, I think that might involve an in vitro fertilization, but in vitro. So let's talk about in vitro. In vitro, basically, when you think about in vitro, you want to think about something. Go away, Facebook. You want to think about something taking place in a test tube. So, in vitro, when you're looking at something taking place in a test tube, you are literally controlling what goes in that test tube. You're controlling the enzymes, you're controlling the ingredients, you're controlling the volume of the test tube. That's in vitro. So you have it's typically a reaction that you want to take place and you want to see a result. And there are all kinds of in vitro assays that are used in medicine, and there are all kinds of in vitro assays that are used in research. And Nicole, I'm sure, has talked about quite a few of these in her live streams. And I know that when I interviewed Nicole for the first time here, we were talking about biomarkers. Well, that's when you are taking a sample from a patient and you're looking for a result, and that result will tell you, for example, why, a patient's blood sugar is too high. So that's in vitro assays, in vitro tests. Oh, yeah. My daughter's a surrogate as we speak. Great to have your presence, Tasha. Yes, great to have you both here. So in vitro, why is this relevant for what I'm talking about? Well, again, my graduate advisor in his lab before I joined, they were able to show that this drug, guanabens, and others like it, were able to cause the protein, our nitric oxide synthase protein, to go away. And their results also suggested that it was by a specific protease. And they came to this conclusion using protease inhibitors. So then his next question was, gotta be careful with my tenses here. What exactly were those drugs doing to our enzyme, and what were the structural changes taking place on our enzyme that was making it degrade? So in order to do that, we had to ask the question in vitro. So I just told you how we were able to create massive amounts of this enzyme here. So we had to figure out how to ask questions about its degradation and its structural changes with the protease using an in vitro system. Okay. So that's what my research involved. So we knew that we could make massive amounts of our protein, and we were good at it. I did lose some sleep over this sometimes because our lab ran on this stuff. But we needed a source of our protease. So the question is, where would we get our protease from? Well, it turns out that this was documented in the literature, and this was done. So all we had to do was look it up. So it turned out that what you had to do was take something called I'm just using this as a visual. You had to take something called rabbit reticulocyte lysate, which basically is what a reticulocyte is. A reticulocyte is basically a blood stem cell. So all of our blood cells, they come from a precursor cell called a reticulocyte. And so what you do is you take that reticulocyte, you homogenize it, you lyse it to make it one big gamish, and then you go back to, once again, your column chromatography. So you have your stationary phase here. You pass your basically mashed up, lysed up reticulocytes over the column. And then what you isolate off is an extract called fraction 2. We called it fraction 2. And then what you were able to do was you're able to put your protein in the test tube with the fraction 2, and you could see if it would degrade or not. And that's essentially what I did in the lab, and that's what I did in the paper I'm going to show you guys. Before I show you guys this paper, and we're almost done, I know this is a lot, I want to show you guys a few techniques, because again, me walking you through the paper without you understanding some of the techniques, it'll just be more gibberish. So one of the, I told you where the proteins came from and how we made them, but how were we able to measure and detect what was going on? One of the primary techniques used, at least during that time, I'm sure science has come a long way in the last 15 years. One of the classic techniques for measuring protein is the Western blot analysis. How does this work? And I'm just going to try to keep this short. What happens is you take your sample, you load it onto a polyacrylamide gel. SDS stands for sodium dodecyl sulfate. That is a detergent. So what that detergent does is it makes these proteins, it denatures the proteins. You see how they're all in their native conformation here. It makes the proteins lay flat and unwind it makes these proteins lose their structure in a nutshell. So you load your protein mixture on the gel, you run the gel, the contents of that mixture, they spread out. And then what you do is you take an antibody. You need a nice antibody. Now, typically, we think of antibodies, since we've been talking about the COVID-19 vaccine, you think of an antibody in terms of detecting a foreign invader or an antigen. Well, years ago, scientists figured out how to harness the use of antibodies as detection tools for science experiments. So what we were able to do was we were able to take antibodies raised against our protein, our nitric oxide synthase, and we were able to detect levels of it. So you take a primary antibody, and then you take a secondary antibody. My secondary antibody was labeled with iodine, so it was radioactive. And then you're able to take a picture like this one here in the corner. Okay, I know that was a lot. I'm gonna look at my comment section here. I wasn't aware that in vitro fertilization could be controlled. Well, yeah, arguably, you have more control over that than in vivo fertilization, Miss Burdine. Okay, so that's a Western blot analysis. You're detecting and you're measuring and you're tracking protein. Okay, so now we're going to walk through the paper, part one of my graduate thesis. I'm going to enable my share screen here. All right, so there we go. In vitro guanabens. Okay, so let me zoom this. Nope, that's the wrong one. Here it is. Okay. Now a couple this is public information, and pretty much anytime a researcher secures funding from the NIH, they're going to publish on their research, and it goes into journals like there are top tier journals and there are lower tier journals, and there are mid majors. My two first author papers were in molecular pharmacology and drug metabolism and disposition. But my point is, this is all available online. And I tried to, in my last science hangout with Nicole, I tried to show some of this, but I didn't have access to the journal. In any case, I do now. So my first author paper for my thesis is entitled Ubiquitination and Degradation of Neuronal Nitric Oxide Synthase in Vitro. Dimer destabilization protects the enzyme from proteolysis. I know that's a mouthful, but I've already explained some of what these pieces are, and I think it'll make sense as we walk through. Okay. So the question is, again, what is happening to this homodimer that is targeting it for degradation by this protease system. So keep that in the back of your mind as I walk through this. Okay. Pretty much every paper, every science paper you read has an abstract. That's a summary of the results. And a very, very low level, I still remember us working together to write this. A lot of work went into the experiments. A lot of work went into writing the paper. Okay. So the mechanism of this selective proteolytic degradation, or in essence, how the nNOS becomes labelized and recognized for ubiquitination and subsequent degradation, has not been determined. So that right there is what we were looking to determine. So sometimes the question is buried right there in the text. I'm going to skip over the intro. I've already discussed these methods at a high level. Okay. So surprise, surprise, everyone. Figure number 1 is a Western blot. Okay. So what you're looking at is a Western blot. And what I did in this set of experiments was I took the NOS protein, the nitric oxide synthase protein here, but specifically, I took the apo form. So if you split this in half, you have a monomer, and there's no heme in it. So when we created it, when we grew it up, it was in a monomeric form. So you think about half of this without the heme, and there was a way that we had to activate it so that it dimerized and it was active. But when I say apo, I'm just talking about half of this without the heme in it. So I incubated that preparation of the protein with the fraction 2 system, which had the protease in it. And that's what you're looking at here. My the incubation went for 90 minutes, so that's an hour and a half. And what you see is that my protein, our NOS protein, it goes away. It's going away with time. So it's getting cleaved up over 90 minutes. As a control, what we did was we boiled the protease system to inactivate it, to see if what we actually were seeing was something biological. And so when we boiled it and labelized it and inactivated it, what we saw over 90 minutes was that our NOS protein was stable. Nothing happened to it. It was there and it was happy. So this was, in fact, a biological process. So in figure 2, we actually quantified it. I quantified it. This is a radio labeled antibody. And so I was able to quantify the amount of protein there using a, I'm forgetting what kind of counter it is. In any case, that's what you're seeing here. You're seeing the protein go away here like this. You're seeing when we boiled the protease system, the protein was stable, right? And then this is the NOS protein without ATP added to the system. So in other words, the protease, it required ATP. So when you deplete, when you don't add ATP to the system, you'll get a lot less degradation. So that's what you see in that top for the white circles. So going back to the cell experiments, they used the protease inhibitor MG132, which is specific for the 26S proteasome. And this experiment is what I've called a dose response. I've discussed this on this channel before. So as you see, as you increase the amount of the protease inhibitor, the NOS protein steadily is more and more protected. So that's a dose response. So this once again verifies that what we're looking at is degradation by a specific protease within the cell. Now, there are other proteases within the cell, And there are inhibitors that would inhibit those proteases. So to check to see if any of the other proteases were active here, I used 3 other protease inhibitors, and those had no effect. So we knew that those were not involved in the process. Okay, I'm going to take a second here and stop. I want to applaud everyone who started at the beginning of this with me and who is hanging on throughout, because I know this is very esoteric, and this is a lot. So thank you to all. Thank you to all. I'm sorry. Thank you to all of you who have hung on. Okay, this system, if a protein is going to get cleaved up, it has to be tagged with ubiquitin. So we wanted to verify and get some clues as to whether or not we were actually looking at a process that involved ubiquitination. So in this figure, what we did was we basically, we had to, because it's a rough extract, there was some ubiquitin still in the system. So we had to drop the amount of the protease system down. So in these experiments, we used 2 milligrams per kilogram, 2 milligrams per ml of the system. And this one, we had to drop it down to 0.2. So we went down tenfold. So under these conditions, we dropped it down tenfold, and we still got 50% degradation. And in this instance, we used an analog of the ubiquitin called ubiquitin aldehyde. What ubiquitin aldehyde does, the reason this is important, you guys didn't even see that. Okay, so dropping the amount of the protease system down tenfold, we were still able to get some degradation. And then we used an analog of ubiquitin called ubiquitin aldehyde. What the ubiquitin aldehyde does is it locks up, it depletes all of this protein out of the system, because it locks up, if I recall correctly, it locks up the deubiquitinating enzymes. So it basically depletes out all the ubiquitin in the system. And once all of the ubiquitin is depleted out of the system, no degradation can occur. And that is what we saw under these conditions. So you add 0.7 micromolar of ubiquitinaldehyde, you start to see some protection of our protein. And then when you add 15, it's completely protected. So this was more evidence that we were in fact seeing degradation by the 26S proteasome system. For the laypersons, micromolar, that's a unit for concentration. And I also, when I was looking at this, these are small differences. And so in some instances, it's important to show statistical analyses. And this has come up when Nicole has been on my shows sometimes, and when I've been in her shows, she's really good at looking at the statistical differences when looking at data, because that's how you prove that what you're seeing is actually real. Okay, I have a few more figures to show, and then I'll be done. Okay, why is that doing that? Okay, the technology. Okay, so with that set of experiments done, we had the system set up, we had it standardized, and we had it optimized. Whenever you're creating, whenever you're using an assay or you're creating an assay, you have to make the determination as to whether or not it's standardized and that it's working. So we thought it was working. So then the question is, so now we can focus just a little bit more, and we can now ask the question, what exactly is happening to our enzyme and to our homodimer that is making it degrade, or that is facilitating its degradation in the rat, in the cell, in the rat, and in the human. So now in this set of experiments, we took the actual homodimer, the holoenzyme, and we incubated it with the protease preparation in our protease system. So similar, let me blow this up for you guys a little bit. So similar to the first set of experiments with the apo enzyme. Sorry about that. Once again, the time course took place for 90 minutes, and we can see that the holoenzyme goes away as well, although to a lesser extent, and we're going to get to that. With the protease inhibitor, it's stable, and quantitatively, we were able to see that as well. So the holoenzyme goes down about 50%, and it's stable with the protease inhibitor. Okay, so then the question is, okay, how is this happening? We were able to run our Western blots in such a way that we were able to look at the dimeric structure in such a way. So when we ran the gels cold without boiling the samples, we still had 50% intact dimer. And that's called an SDS-resistant dimer, which is what you see at the top. The rest of it, we're not sure what it was, but we were able to see some of what was happening to the dimeric structure of the enzyme. So just putting it in the test tube with the protease and letting it go, we could see that over time, the dimer went away. Okay, and we see some of this fraction down here going away. But the interesting thing is when we put the protease in, we see the dimer go away, and we see this band down here start to intensify. And when you quantify it, what you were able to see was the dimer go away in both cases, but with the protease inhibitor, you were able to see the lower band intensify, suggesting that it was converting to something else, and whatever that other species was, it was protected by the protease. So there was a conversion taking place. So then the question is, the next question we asked was, if we stabilize the dimer, the homodimer, can we protect it from degradation by our protease system? And that's the final set of experiments I'm going to show you, and then I'm going to wrap up. So in this final set of experiments, what I did was if you look at panel A and B, panel A and B were treated with this compound over here at the far right, and nitroarginine. And nitroarginine is an analog of L-arginine, which is the natural substrate for the enzyme. So the only difference is you have a nitro group on the end as opposed to this amino group. So in the animal and in the cell, my graduate advisor and that team of scientists that conducted those experiments, they were able to show that this nitroarginine stabilized the protein and protected it from degradation. And so for my experiments, I was able to show something similar, which is what you see here. So when you look at the monomer dimer ratio for the untreated, that one goes away. We saw that before. But for the N-arginine treated, the dimeric structure is stable over time. And for this other species down here, it's pretty much a flat line. When you look at the total protein goes away, but the N-arginine treated is pretty stable as well. When you look down at panels C and D, Let me make sure. Just want to make sure these are accurate. Let's see, B and D. Give me 1 second, guys. Total amount of NOS present. B and D. A. Okay, so the top panel was 7-nitroindazole. Okay, so I misspoke, which was another inhibitor, but it did the same thing. So panel C, that's the monomer dimer experiments again with the N-nitroarginine. The untreated goes away, and the N-nitroarginine for the N-nitroarginine protected the structure of the dimer here. And then in D, this is the total. So the N-nitroarginine protected the protein here. And for the untreated, that went away in addition to in panel D. In addition to the monomeric form here. So the net of it, as Kevin Samuel says, is that these analogs were able to basically hold the structure, to hold the protein together in its dimeric form, and it was able to protect it from degradation. But that's different from blocking the protease from cleaving it up. So this all suggested that this homodimer has to come apart somehow in order for it to degrade, in order for it to be degraded by the protease. So then the question is, going back to the rat and the cell, then the question is, was the drug guanabens here causing the homodimer to destabilize somehow? I'll talk about that in part 2. That'll take another hour for me to discuss. But my initial findings suggested that this structure must come apart in order for the protein to degrade into pieces. The final, I'm going to show you guys this, and then I'm going to stop because it's an hour and 18 minutes. This, I always thought these were pretty to look at. This, what you're looking at, is the ferrous CO complex of the neuronal nitric oxide synthase enzyme. What does that mean? Scientifically, that means that cytochrome P450 enzymes like this have a heme center, represented by this diagram here. The nitric oxide synthases also have a heme center, and that's where the catalysis takes place. Okay. These heme groups, when they're properly coordinated in the active site in this enzyme, and in this enzyme here, when they're properly coordinated, they give off a unique absorption spectra at 444 to 450 nanometers. Okay. And so what we wanted to see was when our active enzyme, we wanted to see if it was losing its heme structure before it was degrading. So what my graduate advisor had me do was he had me run the reaction and then once again use a crude column chromatography so that we could isolate out our active enzyme. So we wanted to see if the heme was still in the protein during the experiments, or if the heme needed to be lost in order for our enzyme to go through the proteolytic pathway? And the answer seemed to be no. So that's the last figure in that paper. And that is part one of my graduate thesis. I know that was a lot. That was probably a lot for non-scientists to understand. It was a lot for the laypersons to understand. But I thought it was important for me to present this because I haven't, maybe in one instance, I presented some hard data. I think I was talking about whether or not chloroquine and hydroxychloroquine were able to stop the replication of the coronavirus. I showed some hard data in those talks, but I hadn't showed any other hard data. So I wanted to do it for that reason. But also, I never talked about how I became Dr. Dunbar. And so there are a couple things I'm going to say in closing about this. This was a learning how to do science was a long process for me. Thank you, Tasha. Thank you, Miss Berdine, for hanging out. Learning how to do science was a long process for me personally. At my HBCU, I'm going to show this blog post. At my HBCU, I was an A student. I was very good at doing the coursework. I was very good at taking tests. I was very good at studying. But going and actually doing the experiments and having to do a research project is a totally different thing. And I remember me and my father talking in a conversation when I was at the University of Michigan, and I was sounding kind of stressed out, and I was stressed out, and I was at my wit's end trying to get these experiments to work and trying to finish. And I remember my dad said, there must be some serious studying. Well, after the first 2 years, it wasn't studying. It was working a research project. It was being in the lab. It was trying to publish papers. It was competing against other labs across the country and around the world. It was trying to make the finding first. That's a picture of me working at my workbench. And the thing I didn't understand when I started this process was that this ultimately becomes an apprenticeship. Just like you could say that it's similar to being the apprentice of a plumber or an electrician or a mason of some sort, because you're working alongside of a master, and the master is trying to get you to be sharp, and the master is trying to train you so that you can go out and do it yourself, and so that you can go out and be productive as a scientist yourself. And me and my graduate advisor, we had a lot of conversations where he had to really hammer me into shape. And there were a lot of times when I could have just thrown up my hands and said, I give up, I quit. He did say up front that he wasn't going to pull punches. And there wasn't anyone else in my family who'd gone through this process. So there wasn't anyone in my family ecosystem that I could go and ask about it. And I just had to tough it out. And so extrapolating out to the entirety of foundational Black America, students who come from our communities, they're usually the first one to do it. So that adds a layer of difficulty and complexity on the process. My graduate advisor, he was Japanese American, his father was a scientist, so he kind of knew what that was when he entered the pipeline. So for me, I wasn't. I didn't have any scientists in my family. So everything had to be learned for the first time. Culturally, when you become a scientist, and when you're on the road to become a scientist, it's a culture change. Being being in science, studying the literature, going to meetings, having to do experiments sometimes, and having to go into the lab sometimes on Saturdays and Sundays. That's a lifestyle. That's, in your mind, you're saying, I'm in school, but, and I'm getting a stipend to do this work, but this feels like a job. And that's why I say it becomes an apprenticeship, because you're learning to do it. You're learning to think. You're learning to plan. You're learning to do the experiments, and you're learning to do science. And it's a craft, and it's a craft. It's a way of life. It's a culture, and that's why one of my critical messages, especially to the boys, is that's why I know it's controversial, but the things that Kevin Samuels is talking about over on his channel, about men focusing in their 20s on their craft and on their careers, that speaks to me, because when I was in graduate school, I got involved in a bad relationship. And this is part of my story, and I'm going to keep telling it. And I didn't know to protect all of this. I didn't know that I had to protect my project, and I had to, not only protect myself and my well-being, but I had to protect my project. And I was a part of a research team. And these research labs at schools like the University of Michigan and Harvard and MIT and the Ohio State University, each lab is a mini company in itself. And the principal investigator is the owner of that lab. And the principal investigator's career is tied to the results and the research in those labs. Just like as a student, your ability to go out and further your career is tied to your own work. So there were times when that relationship that I was in was so chaotic that the entire lab could have been impacted, and the entire lab could have crashed because of my personal life. And I didn't know at that time, I didn't appreciate the importance of what I was doing. I didn't appreciate the depth of what I was doing. I didn't appreciate how the opportunity, but I also didn't appreciate how it was going to impact other people if I let my duties crash. And they came very, very close to crashing on numerous occasions. And that's why when you hear me on the Roger Report, and when you hear me talking to Officer Charles Faulkner, and when I say that I'm fortunate to be here as Dr. Dunbar, that's why. And I think that when you're on a path to success, and you're trying to build yourself, and you're trying to build something, you've got to be a little bit ruthless about your pursuit, and you have to be a little bit selfish about it, and you have to be careful and very, very selective about the people you surround yourself with. In this blog post here, that's me in my research lab, but in the blog post, just briefly, one of my buddies wanted to fight. He almost got into a fight at a nightclub here in Buffalo over a female. My best friend, they probably won't like me telling these stories, but it's to help others. My best friend, one night, he drank a little too much on his birthday, and he splashed somebody with some alcohol, and that guy was following us around the venue that night, looking to exact revenge on my best friend, and I was there with him. So if something happened, it probably would have happened to me as well. I told you about the lady friend I was involved with. All these things could have had an impact on my ability to get my research done and to finish my PhD. And it doesn't stop, because you go out afterwards and you either look to secure a career in academia, or you work in the federal government like I do, or you might go work for a private sector drug company like Pfizer or Merck or Johnson and Johnson. Well, once you become Dr. So-and-so and you're in charge of these projects, and even part of that is just thinking about the science all the time. You don't have time to be thinking about bullshit. You don't have time to be worried about bullshit. That stuff that I mean, there's going to be things that happen in our lives, parents to take care of, children that get sick, health issues. They're going to be things that you can't help. But once you become a scientist, or when you're training and you're in charge of so many things, you can't afford to be involved in bullshit and worried about bullshit. So you have to be very, very selective about the friends you keep, and the intimate partners or the intimate partner you get involved with. So we just crossed an hour and a half. That's part one of my thesis, and I think I'm going to stop here. And let's see what Miss Burdine has to say here. After pushing our way to succeed in various careers, we also end up having to select and find those who has some tolerance for the level reached through it all. Miss Burdine, what does that mean? The last part. Who has the tolerance for the level reached through it all? Yeah, expound upon that. Unpack that a little bit. Going back to what the image guy Kevin Samuels says on some of his live streams, he talks about men should focus on their craft in their 20s. Most science careers, also medicine, you will not reach your earning potential until your late 30s and into your 40s. So I can attest to that. And if I could go back and talk to myself at 25, 26 and 27, I would say, bro, just focus on being the best scientist you can become. Just focus on your craft and the skills. If you're going to be single for a number of years, that's fine. You don't necessarily need to buy a BMW right now. Your job is to get these skills, get these credentials, and get out with the lowest amount of debt possible and with your credit intact. That's what I would go back and tell my 25 and my 26-year-old self. I would tell him, don't, you're still growing into who you're going to be. Don't hang your head because you don't have 100K right now. Focus on the skills, focus on your craft, focus on getting these papers published. You'll be much more powerful in the back end. Ignore the other stuff. And don't get involved with someone if it's going to be harmful to what you're trying to do. So those are things that I would go back and tell myself if there was a time machine and I was going through this process again. So that's all I wanted to say. Hopefully YouTube won't take this down. I didn't say anything controversial relating to vaccines or what's happening now with COVID-19. So I don't expect this to get taken down. There's our answer. Let me see what Ms. Burdine has to say. It means that we sometimes leave our original or beginning level of knowledge and move up to advanced vocabulary and skills that others have to find a way to appreciate, which some will not. Yeah, one challenge is when you ascend, particularly in terms of your familial and your friendship circles. You do grow and evolve in certain ways, and some people will be happy for you. Some people will be envious of you. Some people will be jealous of you. And some of those might be friends or relatives. And you have to, some people will think that you're better. You may be called condescending. And you may just in general evolve past where you are or where you were. And people might want you to stay who you were. And so all of those things are things you have to navigate. The places you used to eat at, you might still go there, but you also might like eating other things. After the whole process, I grew a love for Korean food and Thai food and Vietnamese food. And you just, when you do something like this, you just learn so much. So there's a dance that you have to do once you go back around your own people. And you have to, there's a lot you have to navigate, both personally, culturally, and professionally. Places you used to hang out at, you might think, okay, this is not safe anymore. And there are places that I won't go anymore, that I used to go to, not just in my 20s, but before I ascended and evolved. So there's some places that just I shouldn't go anymore. So, okay. Thank you, Ms. Bourdin, and I appreciate your support on all of this. So I'm going to stop this here. I want to thank everyone who hung on and listened to this. I know how to waterfall content now. So, in every live stream, there is so much meat that you can cut up and chop up into different individual videos. So even just the last part is worth cutting off and creating and making its own video. But for those of you who watched this, you'll probably see parts of this emerge in shorter installments. And for those of you who hung on, I hope you got something out of this. And for those of you who watch on the live stream, for those of you who watch on the playback, if you have any questions, please leave those in the description box below. I'm going to leave my contact info slide once again, in case you want to reach out directly to me. And I'm probably going to leave the links for my those papers I showed. I'm going to leave those in the description box below as well. So there's the Big Words blog site. There is my email that is specific for YouTube, bigdiscuss76 at bigwordsarepowerful.com. I know that's a mouthful. There's the Big Words blog site Facebook page. That's at Big Words Are Powerful on Twitter. And at Anwar Youssef 76, that is me on Instagram. I'm always looking for more Instagram followers, even though I'm not that guy who's taking all kinds of pictures of myself and showing my stomach and me with my shirt off. That's not me. I post a lot of insightful and funny stuff, and then on nights when I think I look really, really suave and cleaned up, I'll take pictures of that. So in any case, everyone, enjoy the rest of your Saturday. Again, if you haven't, please like this stream. And be safe out there, everyone. So with that, as always, remember that your attitude determines your altitude. Look out for live streams on my other channels. And take care, and I will talk to you the next time. Bye-bye.